Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today, the topic is related to biotechnology, and the topic of my today's lecture is the recombinant DNA technology. This is very much important in every field of science, and basically, it is much involved to the biotechnology course. So basically, most of the students are really confused in the different terms that are related to biotechnology, and they are confused in the recombinant DNA technology and the biotechnology and the genetic engineering, these such type of terms. Okay, so today I will discuss and I will make clear that what basically is a recombinant DNA technology, how it is linked with the biotechnology and the genetic engineering. So I will start my lecture in a very simple way, and I will make sure that all the students that are watching this video, they will be clear about the recombinant DNA technology at the last. Okay, so I will discuss it in a very simple way. So I think that it will be understandable by you people. So let's start with the video lecture. First of all, before starting, I just want to give you a very simple and brief introduction about recombinant DNA technology. And in the next slides, I will now then discuss the definitions. First of all, just look to this diagram that what I really want to explain. Okay, so in the modern world, what happens that just listen to the story that we all know that what really fireflies are. Okay, these are such type of insects that produce light in the night. Okay, and in the Urdu we also called it, yeah, it as jugnu. Okay, so these are such type of insects that produce light in the night. Okay, these consist of special type of genes that produce enzymes that are involved in the lightning, in the luminance of this insect. Okay, so for example, this is the DNA of this firefly. Okay, this is the DNA of this firefly, and what happens that this portion of the DNA, or this is the gene that is responsible for the luminance. Okay, this is responsible for the enzyme, for the proteins that are producing light. Okay, so this is we will consider this gene. Okay, now here come to a common plant. Okay, this is the DNA of a common plant. For example, this is a plant. Okay, so and this is the DNA of a common plant. So what really happens that in the recombinant, in the biotechnology, what we do, we basically alter the genes of the alter the DNA of the organism. This means that, for example, this gene is producing light. So what happens, we want to make this plant to produce light. So what we do, we take out this gene. Okay, we take out this gene from this firefly, and what happens, we insert that gene into the DNA of the plant. Okay, for example, now this is the plant DNA, and we have inserted our gene of interest to the DNA of the common plant. Okay, so what really happens, this plant will start producing those proteins and those enzymes, and what will happens, this plant will produce light. Okay, this plant will produce light. So what really happens, that this is all included in the genetic engineering. Okay, so if I again tell you that this is the DNA of the firefly, and we take the gene of interest. Okay, we take the gene of interest through different techniques, and what happens, that we insert this gene into the DNA of the common plant. And what really happens, when this DNA is altered by injecting the gene of interest to it, so what will happen, then this DNA will start transcribing that protein there, and it will start producing light. Okay, so we have also made the plant to start light. Okay, to give light to us. Okay, so in the Japan, what really happens, that you will see these type of plants in the highway, And what happens, they are also looking very much beautiful, and they are also giving light source to the people. Okay, so the light source is transferred from the firefly, or not transferred, but it is all it is we can say copied from the firefly, and this function is also added to the common plant. So then now they are giving benefits to us. Okay, so this is all included in the biotechnology. So I was just discussing this such type of example. So just to make your background, okay, then now I will discuss the different definitions of the biotechnology, recombinant DNA technology, and the other type of terms. Okay, so this is the plant, and it really exists in Japan and every other parts of the world, and they are giving benefits to the people. Okay, so if now we come to the biotechnology, so biotechnology refers to those processes in which living organisms are manipulated, particularly at molecular genetic level, to form useful products. Okay, so this is basically the biotechnology. The light, the proteins that are producing light are transferred from the firefly to the plants, and this is just making giving benefits and useful product to the human beings. So this all field by transferring the DNA to from one organism to the other organism, and making the other organism to produce such type of substance that are giving benefits to the human beings. This is all included in the biotechnology. Okay, so this was all the biotechnology process. Okay, these processes are included in the biotechnology. Now, here come the genetic engineering. So what really happens in the genetic engineering? The deliberate modification of an organism's genetic information by directly changing the sequence of nucleic acid in its genome is called genetic engineering. What really is genetic engineering? That when we altered the normal sequence of the DNA of an organism to a new sequence, so this is known as genetic engineering. We have applied different techniques. Okay, so by engineering, by doing different practices to the to an organism DNA, and making it we can say creating differences to that DNA, that is known as genetic engineering. Okay, and at the last, what is basically recombinant DNA technology? So all the methods that are used in this biotechnology and genetic engineering is known as recombinant DNA technology. Means that we have first we take the specific type of sequence from that organism. So this is basically, we have conducted different methods. So these methods are known as recombinant DNA technology, and then we insert these genes to the other plant and to the other DNA, so by different methods. So these all methods are known as recombinant DNA technology. Okay, so I think now you are clear about the biotechnology, that what is basically a biotechnology, and what is genetic engineering, and what is recombinant DNA technology. Okay, and this is an overview of recombinant DNA technology, that how this process starts. Okay, so for example, this is an organism. Okay, if we consider it as a if we consider it as the firefly. Okay, for example, this is the firefly. It is not a firefly, but just I have given the example of that firefly, so just keep the firefly in mind. Okay, for example, this is the firefly, and this is the DNA of the firefly. So first, what we will do, we will recognize the specific genes that are responsible for the lightning. Okay, after that, we will use different enzymes that are known as restriction enzyme. What are basically restriction enzyme? I will make a video over it, that how it works. Okay, so restriction enzymes are a special type of enzyme that cleave different parts of DNA. Okay, so by using restriction enzyme, we cleaved our desirable genes. Okay, after that, what really happens, we insert those genes into a vector. Okay, and that vector is basically a plasmid, because plasmid are those extracellular DNA components that are actively dividing. Okay, so by the action of actively division, we place the our gene of interest into the plasmid. Okay, and it is also known as a vector. Then what we do, we insert it into our new host. For example, this is the plant. Okay, this was the plant. So it starts from the firefly. We take the DNA from it, the gene of interest, and we inject it into the plant. And after that, these plants are then producing these such a type of proteins. Okay. So now this is the plant that is producing light. Okay. So this is a very simple and a very simple method of recombinant DNA technology. Okay. So these are all involved in it. So hope you have understand and that what is basically recombinant DNA technology. Was a simple introduction about recombinant DNA technology. Hope you have understand it. In the later videos, I will discuss each and every part in detail and what are restriction enzymes, how we place our gene of interest into the vector, and how then the host is producing this certain type of proteins. Okay, so these are separate topics of different lectures. So I will discuss it in detail in the later video. Okay. So hope you have understand the introduction of recombinant DNA technology. If you have any questions, so you can ask me in the comment box. I will answer you there. Okay. Thank you very much. God bless you.